So this is part 3 of the lecture for Unit 1.1. So in the last part, we drew a phylogenetic tree between some different animals where we classified them based on their structures, for example, their jaw and whether or not they had feathers. These are all structures that are big enough for us to see on macroscopic organisms. How do we learn to classify microbes? They're too small to see, and so we need to use other tools to be able to learn about their structures and what makes them up. First, our tool is microscopy. So a microscope will let us magnify the organisms so that they are big enough to see. But even more than then, we use some other techniques. We use techniques to grow them in the lab and learn about how they grow. So we use their properties of growth to classify them. And we are also able to determine what their chemical makeup is. Especially, we're able to analyze, for example, their DNA, their genetic sequence, in order to classify them based on the makeup of their genetic material. So, using a microscope, we're able to classify bacteria based on their morphology, or the shape, size, the way something looks. For bacteria, morphology by itself is not very useful because bacteria in general fall within just a handful of general shapes. Most bacteria are these two shapes on the far left. They're either round spherical bacteria that we call coccus or cocci, or they are rod-shaped bacteria we call bacillus or bacilli. Some other bacteria have some related shapes. So the Vibrio are some rod-shaped bacteria that have a distinct curvature in the rod. Coccobacillus are bacteria that have a short stubby rod shape, and so they look like a combination between the coccus and the bacillus. Then there are two spirally bacteria. So there are Spirillum that have a long wavy shape, and there are spirochetes that have a tight rigid spiral to their bacteria shape. So these are all the general bacteria shapes that we'll see in clinical microbiology. This is the first rapid review question of this part. Make sure you take a minute to answer it on Edpuzzle. So we can also classify microbes based on their properties of growth. So where they grow and how they grow, what nutrients they need to reproduce. If you're a pathologist, pathologists classify pathogens, those bacteria and other microbes that cause disease, based on where they grow and what their growth looks like when they present in patients. So microbes can be grown in the laboratory in several different conditions, and we can characterize their growth in a laboratory setting. This is especially useful for differentiating between a lot of common clinical microbes, as we'll learn in lab. We can also classify microbes by where they grow in the environment. Many microbes grow inside of other organisms. For example, the rumen or the gut of a cow is a rich source of lots of different microbes. We can also classify microbes based on their genetic sequence. So one of the tools that we'll use in clinical microbiology is to classify bacteria based on the sequence of a gene that is part of what's called the ribosome. So the ribosome are complexes within cells that make cellular proteins. They're some of the biggest molecular complexes inside of cells. Their three-dimensional structure of these molecules looks like this. They have two big halves. They have a small subunit and a large subunit. And both of those subunits are made up of a number of molecules that associate with each other and let this little tiny machine do the things that it does. So as I said, ribosomes make proteins, and this is one of the most essential functions in all cells, right? And we know all life on Earth is related, and in all cells, the things that make proteins, the things that are essential for those cells to live, are ribosomes. And so ribosomes are an example of a homologous structure. They are complexes that are similar between organisms because they have an evolutionarily related origin. So ribosome genes are really useful for classifications because all organisms have ribosomes, and because they're so important, the sequences haven't changed as much as other genes throughout evolutionary history. And so the 16S ribosomes of lots of different organisms, 16S ribosomes, have very similar sequences in some places, and then slightly different sequences in others. And so we can distinguish between different prokaryotes by using the common sequences to compare them and the different sequences to differentiate between them. Those common sequences are called conserved sequences. It's when the gene sequences are similar across lots of different species. So we can use these conserved sequences in the structures of genes to compare different organisms. So if we look at a DNA nucleotide sequence between a group of different evolutionary related organisms, we can see that this conserved sequence, which is just 6 nucleotides long, in many of those positions on the sequence, the identity of the nucleotide, of the chemical that makes up this DNA, is the same. But on some positions, there are some differences between the organisms. We can see that species 1 and species 2 both have a change. They have a C at position 3 instead of an A, which all the other organisms have. So this is an indication that these two organisms have a structure of their gene, a sequence that is more similar to each other than it is to other organisms. Species 3 and species 4 have a different shared change. They have a G at position 5. That might suggest that these two organisms are more similar to each other than they are to other organisms in this group. So this technique of using these sequences, specifically from 16S RNA, was pioneered by two scientists named Carl Woese and George Fox, who studied the sequences for hundreds of micro and macroscopic organisms, and realized that a lot of the assumptions that we made about microbial evolution were wrong. And so in modern microbiology, we now use sequences to classify different organisms. So think about how we classify and create phylogenetic trees and the sequence that we just worked with. See if you can select the best phylogenetic tree that represents the evolutionary relationships between these organisms the best. We can use these sequences and phylogenetic relationships for practical purposes. For instance, phylogenetic trees are one of the reasons that we know that SARS-CoV-2 spilled over from bats at the beginning of the COVID-19 pandemic. This is a phylogenetic tree drawn from DNA sequences that were sequenced from here. These are all sequences from patients who were treated in the early stages of the pandemic. They had COVID-19 disease in Hunan, China. All of these sequences are nearly 100% identical to each other. They are related to, but somewhat distantly from, the SARS coronavirus sequences. SARS coronavirus is the virus that caused the original SARS epidemic in 2004. They are also related to, although somewhat distinct from, the MERS coronavirus. And so these are viruses that have caused a couple of different outbreaks throughout the world. Right. So SARS-CoV-2 is similar to these other two SARS coronaviruses that have been studied by humans in labs. But the most similar coronavirus that has been identified was one that was taken from a wild bat in Zhejiang, China. And that sequence is 98% identical to SARS-CoV-2, the virus that causes COVID-19. And so we know that the sequence of SARS-CoV-2 is most similar to viruses that were found in wild bats and less similar to other human viruses that have been previously studied by researchers in labs. Right? So the virus is not similar to the laboratory viruses that we work with, and it is similar to a virus found in wild bats. We know because of the loss of habitat for many of these bat populations that their interactions with humans and human livestock are becoming increasingly frequent. So this might be one of the reasons why we've seen these spillover events from these bats that harbor lots of these coronaviruses that seem to be good at making us sick. So microbes are the most abundant form of life on Earth. They're all over the place, from boiling hot springs in Yellowstone National Park to the freezing ice of Antarctica. And they come in lots of different shapes and sizes and varieties. These are several different examples of types of microbes that we'll talk about in this course. We have some bacteria up here, E. coli cells. We have some fungi or molds here in the sporangia. We have single-celled eukaryotic animal parasites. It's like this Giardia that causes intestinal disease. We have single-celled archaea, which are these interesting microbes over here. We have viruses like HIV, which is shown here in this electron micrograph, infecting a human CD4 positive T cell, one of the cells of our immune system. And we have some microscopic algae that live in the ocean. So in this course, we're going to focus exclusively on microbes that impact human health. Microbes have an incredibly important role in the environment and in the broader scope of the planet. But this is clinical microbiology, and we're going to talk about two types of organisms. We're going to talk about pathogens, which are microbes that make you sick. We're also going to talk about beneficial or commensal microbes. These are microbes that live in our bodies at all times, make up what's called the human microbiota. The human microbiota, we're just beginning to really get a good sense of what the human microbiota is and how much of an impact it has on our health. What we now know is that about 80% of the cells in the body, in your body, are not human cells. They are cells of bacteria or fungi that live inside you and contribute to the chemical makeup of your person. And so we're learning a lot of new stuff about the human microbiota and its impact on our health. And so we'll talk about it, the good and the bad, throughout the course. So these are some of the microorganisms that we'll discuss in this course. We'll talk about bacteria, which are single-celled organisms that are prokaryotes, so they don't have a nucleus. They live almost everywhere on Earth, including in the human body. There are millions of species known, but less than 100 are human pathogens. These human pathogens have a huge impact on human health, and so we're going to talk about those a lot. But know that the vast majority of bacteria are, on the whole, better for us than bad for us. There's over 1,000 species of different bacteria just in human bodies that make up the human microbiota. Archaea are prokaryotes. They're similar to bacteria, but are not bacteria. In fact, if you look at their chemical makeup, they are quite distinct from their bacteria prokaryotic relatives. They're more closely related evolutionarily to us than they are to bacteria. What's interesting about archaea is that there's no known pathogens. Right? None of the archaea that we know about cause disease in any animals. But they are present in the microbiota of us and many other animals. Viruses are, for our purposes here, not considered organisms because they are not made of cells. They lack many of the essential functions that cells have that are required for cells to reproduce. They need to infect a cell to reproduce their viral structure. Viruses do cause a number of human diseases. They cause COVID-19 disease, influenza, HIV, and Ebola, which is the virus pictured here. We'll also talk about eukaryotic microorganisms that fall into a few distinct categories. We'll talk about fungi. Fungi are both pathogens and part of the human microbiota, and they are also important for nutrition. We use yeast for making a number of products and also eat mushrooms. Protozoa are single-celled eukaryotes. There are many important pathogens in this group, including malaria, giardia, which is pictured here. Helminths are multicellular parasitic worms. As you can see from this picture here, a guinea worm being pulled out of an infected wound, they're not actually microscopic. They're often big enough to see with the unaided eye. But their diagnosis in many settings is done by a microbiology lab, and their identification of their eggs in an infected person.